The 29th. So let's see if we can fill this first part in. Mutations are blank in the structure of blank within the blank. Mutations are changes. And what are we changing? The structure of DNA. And then DNA, where is it located in the cell? Within the nucleus. You guys are awesome. Most excellent. DNA are changes in the structure of DNA. Going back, here's our structure of DNA. So for example, here's our phosphate group attached to our sugar group. What happens if I erase this? Could that be a mutation? Absolutely. It's changing the structure. It's changing the structure of our DNA. So let's think we have two causes of mutations. One are spontaneous. Spontaneous, they occur spontaneously, meaning that the DNA is just not replicated correctly, right? Nothing necessarily caused it, it just happens. The process of making DNA and the process of transcription, translation, you remember we learned about those processes? They're very fragile, meaning there's a lot that can go wrong. So this happens when DNA is simply just not replicated correctly. When DNA is not replicated correctly. Our second cause of mutations are called induced. Who can tell me what induced means? Let me give you guys a hint. Here's an example of something that can induce mutations. That's very bad for you. Smoking. Another thing that can induce mutations, a cause, would be you guys know this symbol? Radiation. Radiation, absolutely. Alyssa, do you have
kind of like mutations that are caused because of something that you did or something that you were exposed to?
Totally, something that you're exposed to that causes a mutation. What else, Xander?
I kind of just want to say that cigarettes are a form of radiation because they cause radiation inside you.
Absolutely, right. And they're a carcinogen. Can cancer be a mutation?
Yes.
Yeah.
Totally, right? Absolutely. So not all mutations are equal. They vary on how severe they end up being, right? So we have the first type that is called point mutations. Point mutations. And these are mutations that only affect either one gene or less than. So they affect a small portion of the DNA. Affect a small part of the DNA. And then up underneath here I'll write up to one gene. Up to one gene. So the most important thing to remember with this is that it's not affecting a whole chromosome. It's just affecting a small part of the DNA. So these, right, you might have a mutation that's a point mutation and you might see no effect from it. It might not even change your phenotype or your characteristics of the trait that that gene codes for. Then the large-scale mutations, you can write large-scale mutations or you can write chromosomal mutations. And this is when you are affecting the structure of a chromosome. So on each chromosome, how many genes do we have? Do we only have a couple or do we have a lot? We have a lot, right? So if we can switch, or if we end up having a chromosomal mutation where quite a few, right, a whole part of the chromosome is changed, that can have quite a large effect. So then our type of point mutations, let's add point in parentheses right there. So that way remember we're drawing the point mutations. We have 3 types. The first one is called substitution. The first type is called substitution. I know, it's crazy, it just happens. Substitution. So what this looks like on a DNA strip. Let's go ahead and draw it. Let's draw 4 base pairs. And then on the bottom, let's say right here, we have an A. Which nucleobase is supposed, or nucleotide is supposed to bind with A? T, right? Adenine goes to thymine. But what happens in substitution is that thymine is substituted and there's a C put there instead. There's a C put there instead. Or there could be a G put there instead. This is when this is substitution. Our first type of point mutations. And 2 things can result from this. Still see that? Excellent. I'm going to draw 2 arrows off of this. 2 things can result from this. The top one is that nothing bad happens, or nothing changes. Because our DNA has a system of checks and balances. So there is an A on the bottom, and then it recognizes, oh, there's something wrong, these base pairs don't go together, so it goes back through and it takes the C out, and then it fixes it by putting the correct base pair in, thank you, and it puts a T in. So it recognizes these don't go together, takes the C out, and then puts a T back in. So it fixes itself. C leaves, T goes back in. The second thing that can happen, and this is where changes take place. What if it thinks that the C is the correct base, when actually it's not? That's the one that was substituted. And it ends up keeping the C, and then which base pairs with the C? G. It ends up putting a G, and then gets rid of the A. Uh-oh, yep, exactly. Because remember, how many bases code for an amino acid? 3, right? So every 3 codes for an amino acid. So let's say this one is a G, and this one's a C, and it's supposed to be G, A, C, and then we end up getting G, G, C. That will be a different amino acid than what it's supposed to be. So then our proteins are all going to change, and they'll be different because of it. That's how mutations take place. The second type of point mutation is called insertion. What does it mean to insert something? Insertion. Jack, what is insertion? What do you think?
When you add a gene inside.
You add it inside, absolutely. Right, so if you go to the vending machine, you insert your dollar bill into the vending machine.